years I've been focused on biological sciences. There's some of the insights that come from biology, but also the methods that are used in science to obtain those insights. So this exhibition, in a way, I think, reflects my interest both in the wonders of science, as Professor Brian Cox might say, and also something around the methods that are used in science. And these three kind of installations are particularly focused on that. But this one, which I call Trillion Superheroes, which is around bacteria, so bacteria and antibodies that are in our bodies for fighting. I remember when I was a lad, when we did studies of antibodies, you used to get, I don't know, most of you are much younger than me, so you probably won't relate to this, but you occasionally saw black and white granular movies of these kind of fiddly little cells that kind of fighting off fiddly little bacteria, all granular and uncertain. And that's the image I had for most of my life about these processes. And then about 2 years ago, I came across a book which was based on photographs taken from an electron microscope of these tiny little objects. And these are scanning electron microscope where you don't have to dice and slice things, kill them off before you put them in the microscope. You can actually see the real life specimen and it scans it. And there were these fantastic colour photographs of these cells within our bodies, the white blood cells that fight off infection in our body. And they're really incredibly colourful and incredibly strange, very kind of alien-like quality in them, which attracted me. But I was very frustrated because it was like they were kind of very flat images, and I couldn't, there was something inside me that wanted to turn them into three-dimensional objects. I kind of thought, no, what would you like? These kind of they were real things that you could sort of almost like go around. So I decided that's what I would do, which was to recreate them as in wax, as those kind of strange cells. I also then started doing a bit of reading around the subject, and I discovered that it wasn't just one sort of antibody cells. In fact, there's a whole ecology of these cells, and there are trillions of them inside our body that work together to fight infection, fight bacteria. And of course, I couldn't cover the vast sort of pantheon of those cells, so I decided to kind of focus on 5 different cells, the 5 different types of cells, antibody cells in the body that have different functions in terms of fighting bacteria. So there are something called macrophages, which go around. Now, there'll be loads of you who know a lot more about the subject than I do, so I'll be extremely embarrassed if you were to point out that I'm wrong. But this is artistic license, right? This is simplification of the process. So, so the way I kind of saw it was that there was one set of cells that wandered around the body, picking up markers from harmful bacteria. So there's kind of some things that called antigens that cover bacteria. So they kind of pick them up, pick up markers, and they go to another type of cells, which are kind of T cells, which say, here's a harmful bacteria, here it is. And these cells pick up these markers, and they become hunter-killer cells. And it's not a dramatic use of the word, this is what scientists use to describe the transformation of these cells, otherwise kind of, generic cells, into hunter-killer cells. They take up these antibodies and then they go off and look for those dangerous bacteria, and they zap them. But they don't stop there, because these cells also produce what's called memory cells, so that the body always remembers the infection that had fought. So there seems to be this kind of really complex ecology, and these complex shapes of cells and things that I really got into. I really wanted to kind of turn them into sculptural processes and installations. And the final component of this was that when scientists study these tiny little things, what they do, because if you imagine, they are, well, as I imagine, they're tiny little jelly-like, almost transparent things in your blood cells. What scientists, as I understand it, do is they use fluorescent dyes, specially constructed fluorescent dyes, to mark these cells. And they don't mark the cells themselves, they also mark special structures in the cell. So different fluorescent dyes interact with different structures in the cell, so you then get the shape of the cell slowly forming. And the shape is formed by the fluorescent dyes. So that gave me the clue as to how I could mimic that scientific process in terms of using fluorescence to mark that particular process. So for those of you who've seen in there, that's what you've seen, which is a kind of, I hope, what you've seen is a interactive installation with lots of kind of components doing lots of different things and fluorescing like mad inside, because in a way that's how I imagine, you know, in their heads, that's how kind of scientists see that kind of particular process. But this is a simplified version of it completely in my imagination.